Aging-related cognitive decline serves as the standard biological baseline for most modern populations. Data from an observational study at the Barrow Neurological Institute followed participants over 4.5 years. Those who strictly adhered to a specific nutritional protocol showed a 53% reduction in the risk of developing Alzheimer's disease. This protocol, known as the MIND diet, synthesized specific elements of the Mediterranean and DASH diets. It prioritized a narrow group of nutrient-dense foods, like these fresh berries, as primary sources of botanical antioxidants. The data reveals a clinical gap. While dietary adherence shows measurable success, billion-dollar pharmaceutical trials targeting brain plaques have repeatedly failed to produce similar outcomes. Identifying how these dietary inputs protect the brain requires discarding outdated models of Alzheimer's pathology. These protocols function as targeted biochemical therapies, addressing the underlying network failure that precedes cognitive decline. At the macroscopic level, the pathology of Alzheimer's disease is defined by a progressive, severe structural loss of brain volume. At the microscopic level, researchers identified two pathological hallmarks: extracellular amyloid beta plaques and intracellular tau tangles. For decades, the amyloid cascade hypothesis suggested a strict linear progression, where amyloid beta accumulation directly caused tau tangles, which then triggered cell death. Clinical outcomes have largely invalidated this linear path. Removing the plaques alone has not halted the progression of the disease. Modern pathological models indicate that amyloid and tau do not act in a simple sequence. Instead, they act in parallel, driving a synergistic feedback loop. This dual interaction silences cortical neural circuits significantly faster than either single pathology could ever do on its own. Treating this bidirectional cellular siege requires a chemical intervention that can stabilize the entire system simultaneously. The process begins at the neuronal synapse, where the amyloid precursor protein, or APP, protrudes from the cell membrane to manage normal cellular functions. In a diseased state, enzymes called base-1 and gamma secretase clip the APP at incorrect sites, releasing smaller peptide fragments. These peptide fragments misfold and clump together, forming toxic complexes known as amyloid beta 42 oligomers. These diffusible oligomers are the primary neurotoxic species, capable of spreading throughout the brain. Eventually, these oligomers aggregate in the extracellular space to form the large, insoluble plaques visible in late-stage Alzheimer's. Extracellular accumulation is only the first half of the pathology, acting as a toxic catalyst for an internal cellular collapse. Inside the neuron, a secondary crisis unfolds along the axonal microtubules. These microtubules act as internal cargo transport tracks, stabilized by interlocking supports called tau proteins. This railway system is managed by regulatory enzymes, kinases, which attach phosphate groups to the tau, and phosphatases, which remove them. Systemic stress and inflammation hyperactivate pathogenic kinases, notably CDK5. The rogue CDK5 hyperphosphorylates tau, forcing the supports to detach and leave the track vulnerable. Detached tau twists into dense neurofibrillary tangles, while the unsupported tracks crumble. This internal structural destruction halts all synaptic communication, completing the lethal cellular siege. These extracellular amyloid and intracellular tau pathologies are mechanically linked across the network. Damage spreads through a remote interaction, occurring directly across the synaptic cleft. Extracellular amyloid beta oligomers physically bind to postsynaptic receptors, specifically TMEM97. This docking triggers internal cascades that drive localized neuroinflammation and excitotoxicity. To propagate the disease further, neurons utilize tiny transport pods called exosomes. A diseased presynaptic neuron packages toxic amyloid and oligomeric tau into these spherical vesicles. The exosome crosses the synaptic gap toward a healthy neighboring cell. It then fuses with the postsynaptic neuron, injecting toxic tau seeds directly into the new environment. The surrounding amyloid beta actively accelerates this exosomal tau seeding and propagation. Breaking this feedback loop requires chemically stabilizing the postsynaptic membrane to prevent the initial receptor binding. This stems from a structural reality. The human brain is composed of approximately 60% fat. Docosahexaenoic acid, or DHA, integrates directly into the lipid bilayer of the neuronal membrane. A high concentration of DHA fortifies the synapse, physically blocking toxic amyloid oligomers from docking at the TMEM97 receptors. Simultaneously, eicosapentaenoic acid, or EPA, works to suppress local neuroinflammation. Fatty fish intake serves as a mechanical reinforcement of the neuronal membrane against the synergistic Alzheimer's loop. The MIND diet also relies on the consumption of leafy greens, olive oil, and berries. These foods neutralize the oxidative stress generated by free radicals in the aging brain. Unchecked oxidative stress is a primary driver of the Alzheimer's pathology model. Specifically, it hyperactivates the kinases that destroy the internal microtubule tracts. The MIND diet provides vitamin E and botanical antioxidants that physically collide with free radicals, neutralizing them before they can trigger internal damage. Quenching this stress represses CDK5 activity, allowing structural tau proteins to remain securely fastened to the microtubules. These antioxidants act as organic kinase inhibitors, intercepting the biochemical trigger that leads to tauopathy. The resulting neural network is structurally fortified by neurolipids and chemically buffered by antioxidants. Alzheimer's is a synergistic failure, and fighting it requires a multi-target defense. Modifying dietary inputs provides the specific biochemical building blocks required to intercept the cellular collapse. The 53% reduction in Alzheimer's risk correlates with these specific mechanics, indicating that the brain's resilience is a direct product of its dietary inputs.